not for the audience. Okay. So, excuse me for the very long title. I actually do not remember my title. It was a last-minute change right before our publication came out. The editors thought our title was too scientific and not catchy enough. And so they made up the title. So basically, I'll be telling you about very biological, functional problem that we have been working on. Completely coincidentally, I didn't realize how mathematical the audience would be. And I actually cut out all of the mathematical equations and the computational part of my talk. So sorry for that. If you're looking forward to the details, you can just come and talk to me afterwards, okay? So I'll be telling you about human-induced pluripotent stem cells, iPSCs. And in particular context that we're interested in are differentiated iPSCs that are more like liver cells, hepatocytes in particular. This is because we are very interested in cardiovascular diseases, which is one of the leading causes of death, not only in the United States, but also in the world. My name is JoSon, by the way, if you didn't see that. So I'll be telling you a little bit about how to find statistically regulatory variation associated with complex traits. And I'll be telling you a little bit about how that modeling worked, both molecularly and computationally. And I'll tell you a little bit about some of the exciting results we have from the molecular side of our collaboration. So just to give you a little prompt, the GWAS era, we think has been a great success. The observation, however, is that the majority of complex traits is explained by common and non-coding variation that we know very, very little about. And during the course of this retreat, we have been hearing a lot about the non-coding and epigenetic regulation studies that a lot of people are working on, and that is one of the biggest problems that a lot of people are focusing on. Because we know that statistically a lot of these things are important, we just don't know why or how to explain it. So the hypothesis that we are working on is that many GWAS variants function by modifying gene expression, for example, that has a cellular phenotype changes, which ultimately would lead to organismal phenotype changes. Now, of course, there are a lot of problems associated with it, and one of the examples being that the regulatory elements don't just work with the variants that are right next to them in the genomic space. They sometimes work in large distances. Not only that, there are other biological phenomena such as linkage disequilibrium that make it very, very difficult to statistically and computationally tease these things apart. Now, I do work on a lot of projects that involve many aspects of these problems. The one in particular that I will tell you about is actually kind of solved and has been published on. So the ultimate big picture question is actually to try to find causal variants and ultimately causal genes. And if you're lucky enough, maybe lead to some kind of therapeutic interference that you can work on that actually has biological consequences and help people, for example. Now, before we get to that, one of the problems that we started focusing on a few years ago, Eleazar is a part of this, is that one of the major issues is that finding causal genes is not as simple even when you have a lot of data. You have to have the right data, one of which being a particular cell type or tissue. So for example, if you're studying cardiovascular disease, your interests are probably, for example, cholesterol levels that is related to liver, or heart tissues, if you have access to heart tissues. Sometimes it's very, very hard to get them. And I'm showing you an example of a validated functional study that has been replicated in many aspects, including animal model studies and human, what we call human knockout studies that have de novo mutations of stop codons of particular genes. The example is called sortilin-1. It was first published based on association studies that are purely based on statistical significance. But then afterwards, it has been validated in vitro studies, in vivo studies, as well as in human studies. And there are several directions that this is moving forward, including therapeutic interventions. Now, this actually is a very interesting example because sortilin-1 has a very liver-specific expression levels. So if you did not study liver, you probably did not find it. And that was the case in a lot of the candidate genes. Now, of course, I'm here as a part of the GTEx consortium, Genotype Tissue Expression Project. We aim to ascertain up to 960 individuals and up to 55 tissues per individuals. These are postmortem tissues. And we want to characterize and catalog RNA sequencing and whole genome sequencing of these individuals, and one of the goals being, for example, whether gene expression can tell us the difference between a brain and a heart, for example, broadly speaking. There are several other consortia that are working on different aspects of genomic, epigenomic, and transcriptomic data. Phantom being one of them, another one of RNA-seq consortia, ENCODE and Roadmap Epigenomics being a famous example of epigenomics consortia that has a variety of biochemical assays and different sequencing data that are publicly available for a lot of other people to do their research on. So, one in particular I would like to advocate today to you is induced pluripotent stem cells as a model system. There are several advantages of this that I will cover later part of this talk. This is a part of another consortia called Nextgen. Nextgen basically aims to create a mass assembly line of iPSC generation, which traditionally has been very, very hard and very, very expensive. Just to be brief about it, they have succeeded somewhat. And has been generating thousands of iPSC cell lines that have been differentiated into different cell types that resemble primary tissues that we would obtain from humans. And one of the advantages of iPSCs that they're advocating is that it's replenishable, by which I mean instead of getting biopsy of a liver, you can get a blood sample, which is fairly non-invasive compared to other biopsies. And then you can reprogram those cells and then turn them into something that you would be interested in for your own study. So just to give you a little outline in a cartoonic manner, you have some individuals that have particular trait you're interested in, perhaps a disease. You run GWAS or some sort of statistical test that allows you to look at genome-wide variants, for example. And in this particular example, I'm pinpointing GLGC, Global Lipids Genetics Consortium, I think. Sorry, blanking on the details. And you have a bunch of regions that you find interesting based on the particular trait of your interest. Now, you have perhaps the same population cohort or a similar population cohort that you can actually get biosamples from, for example, whole blood. And you do a lot of in vitro assays, including adding some factors that I actually do not really know of. And then you turn them into something that you want, for example, induced pluripotent stem cells that have pluripotency to turn into practically anything that you want within the human body. And then you differentiate them by other biochemical means into something that you would like to study, for example, hepatocytes for liver. And then we actually did a lot of quality, well, we as in Royal We, I didn't do wet bench work, but we did a lot of quality control work and positive and negative control comparisons in order to figure out whether these hepatocytes really are representing the primary hepatocytic tissues from liver samples, for example. Now, ideally, you would carry this on, phenotype them, characterize them, perhaps incorporate them into other studies, including one of the latest would be the therapeutic incorporation of the iPSCs into humans, which has been done for some very, very advanced medical studies. Ours do not go that far, but we do have animal studies that validates a lot of our functional findings. So it suffices to say that, yes?
Does the cohort overlap with GTEx?
Sorry?
Does the cohort overlap with the GTEx cohort?
No. That's actually a very good question. So, well, I am not 100% certain. So a lot of these individuals that are incorporated into GTEx are organ donors or some sort of individuals that are that have agreed to be a part of a research. So we know for a fact that, say, a handful of individuals that are in GTEx happen to also be a part of ENCODE, for example. This is after fact, right? And because of the privacy issues and security concerns and a lot of different things involved in how research subjects become research subjects and distributed, we don't actually know details of whether these individuals ever were incorporated into Nextgen, for example. But as far as I can tell, they do not overlap. So just simply stating that developing this model system has been one of the biggest parts of this effort, and I'm just showing you a very, very simplified skim of how iPSCs would become a differentiated, relatively mature hepatocytes that we would study. It takes approximately 20 days. I would just tell you that it takes a lot longer than 20 days. One of my collaborators has been working on developing this protocol and optimizing it to apply it to hundreds of samples over several years. But at the end of it, good thing for me is that we had about 90 iPSCs, and almost all of them has been differentiated into iPSC hepatocytes that are sometimes referred to as HLCs, or hepatocyte-like cells. And we do have primary hepatocytes that are from livers and has been sorted out to contain positive controls for our assays. And we do have GTEx whole liver samples that are equivalent in number. And this was very important. This was actually very important for statistical analysis. So a lot of the iPSC cells in previous studies have been in numbers of dozens. And that's actually very, very difficult to have any statistical power to run any tests. And so one of the keys that led to my collaboration with these collaborators was that we finally had a large enough number of samples that have been generated and characterized that we could actually start doing some computations on. So we have whole genome sequencing and genotypes, and actually for some of the individuals we even have, for all of the individuals we have whole RNA sequencing data. We do have genotype from all of the individuals. For some of the individuals, we even have whole genome sequencing, but that is not a part of the study that I'm talking about today. So for the rest of the talk, I'll be mainly talking about comparisons between the iPSCs, HLCs, and the whole liver samples. So one of the things that we are very, very interested in are looking at genetic variation that has both statistical and functional associations to something that we can actually measure, for example, gene expression. So one example would be EQTLs, or expression quantitative trait loci. So we are looking at, for example, addition of a minor allele on a SNP that seems to have an effect on gene expression increase or decrease, for example, that we can actually validate on in molecular settings, and that we can actually see some kind of large enough statistical effect size and significance that we can actually detect on a large scale. Another example that I won't really talk about today is called allele-specific expression, or ASE. It basically is pretty much similar to EQTLs in a sense that we are interested in genetic variation that has molecular consequences, for example, gene expression change. But in this particular case of ASE, it actually detects the difference between different strands of a DNA. So it has a proven statistical power for rare variants and smaller subset size samples. And that was of interest to us because compared to something like GWAS that has hundreds of thousands of individuals, our study only has a couple hundred individuals. So we are exploring different aspects that would give us more statistical power. Suffices to say, morphologically and biochemically, our HLCs were quite similar to hepatocytes, and that was comforting to us. It was a positive control. And one of the first things we did were basically clustering the gene expression levels of differentiation markers, or sometimes we call them pluripotency markers. So these were not discovered by us. These were discovered by other individuals that study embryonic stem cells and iPSCs in different contexts. And they have discovered that when they compare those cells to mature cells from primary tissues, there are about 50 or so genes that seems to have very distinct patterns of gene expression between those stem cells and non-stem cells. Of those, about 23 of them are actually relatively well expressed in our samples. So we use them to see the patterns, mathematical patterns using principal component analysis within our data sets. So here, if I can move. Most variants for differentiation markers that we used in this QC step were explained by PC1. That huge bar on the left side is PC1 explaining approximately 75 to 78, I think actually. Yeah, 78% of the variants that we see within our data set. The rest of them are fairly trivial, PC2 being approximately 7%. So we got curious about what this even means and whether we can explain any parts of this. I mean, a part of it is that we knew that these were differentiation markers. So we are hoping that we will be able to validate that these would show the difference between the primary tissues we have as positive controls versus the iPSCs that we have in our data set. So yes, indeed, PC1 being the x-axis and PC2 being the y-axis, we do see a huge distinction between iPSCs and everything else that we tested in our PCA. Those are primary hepatocytes that I have mentioned in the beginning, 3 primary hepatocytes that we have included for positive control. Those are GTEx liver, whole liver samples and validation livers that we got from another set of collaborators. And those are iPSC hepatocytes, meaning PC1 is distinguishing between stem cells and non-stem cells, and PC2 is distinguishing between mature primary cells versus relatively immature differentiated iPSCs that are hepatocyte-like. So that was fairly comforting to us. It was another level of positive control, and we repeated a lot of these similar experiments using different clustering techniques, different mathematical means, to try to figure out whether what we are looking at or trying to look at that are hepatocyte-like, we think, are indeed representing the primary tissues that we want to study ultimately. So,
What is the PCA on all the genes?
on all of the genes, it's actually quite similar, except HLCs become a little more sparse, and you start to see the sub-pockets of HLCs. And when I asked my molecular researcher collaborators about those, so what's amazing about these collaborators is that when I mention a sample name, they know, oh yeah, that's this cell line that we differentiated on this month or something. And so they tell me the properties that they know about these. And not all of them are explained, but for some of them at least, apparently for iPSCs, molecularly speaking, some of them differentiate better than others, and some of them mature better than others. I don't actually understand the details very well, but according to my collaborators, some of these that seem to subcluster within the HLCs tend to be a poorer differentiator. So they have all the characteristics that we expect to see in HLCs, molecularly speaking. But when you compare them side by side with ones that are more hepatocyte-like, those things tend to be a little less differentiated, less well differentiated, is the best way to put it. We do have a lot of other positive controls that I'm not going to talk about in a whole lot of details. So basically, we did a lot of computational analysis trying to compare our HLCs to what we know liver cells to have. So for example, we looked at a bunch of liver-enriched genes and whether they would show up in our HLC cohort, and yes, they do. It was also true when we ran pathway analysis. A lot of the liver-related pathways did show up, which was a positive control, but nothing particularly novel. And so we finally got to the eQTL scans of genome-wide, transcriptome-wide data that we have for iPSCs, HLCs, and the whole liver samples that we have from GTEx. We have a bunch of hits. We weren't particularly interested in any hit. We're interested in the hits that we could actually validate, functionally speaking. And so we looked at, sorry if you hate this sort of diagrams. I actually don't really like the diagram, but it's the best way to show this sort of numbers. So when we do genome-wide eQTL mapping and have very general cutoff of false discovery rate 5%, we end up with something in the order of 2,400 e-genes, e-genes being a gene that has any genetic variation that is statistically significantly associated with a gene expression level, okay? So for HLCs, we have about 2,400 of them. iPSCs have more of them, 3,600. And GTEx livers, we found about 1,500. Well, actually, yeah, the number dropped a little bit after I fixed the annotations. But something in the order of 1,500. We are actually interested in cell type specificity, for example. So one of the reasons why we had iPSCs, HLCs, as well as primary tissues from GTEx was that we are actually interested in the genes that will be HLC specific rather than iPSC and iPSC hepatocytes, for example. And so we ran meta-analysis across cell types from Eleazar's lab called MetaTissue. Basically, it incorporates mixed effects models and looks at posterior probabilities that a certain signal came from one study over the other. Is the simplest way of saying it. We did that and found a bunch of things that indeed has higher posterior probability of being cell type specific and not to the other cells. We also wanted to look at very trait relevant regions. And so we did some preliminary comparisons to the other GTEx tissues, the other 43 that have been published and are available online, to try to figure out whether they're liver specific or would show up in any number of tissues that we are less interested in this particular context. Tissue relevance. We also wanted to know whether they have colocalization of cellular and organismal phenotypes, by which I mean we happen to have summary statistics of GLGC or lipid cholesterol level studies that are showing statistical significance of particular genetic variation and its contribution, statistically speaking, its contribution to a complex trait such as LDL cholesterol, total cholesterol, triglyceride levels, et cetera, that are very, very relevant to a liver phenotype. So we used a statistical method called colloc that basically looks at, well, since Brian has mentioned base vector, I can say base vector, that basically looks at base vector and the probabilities that a particular GWAS signal would coincide with a cellular phenotype, which is EQTL signal, right? We did that, and we found a set of E genes that are actually very interesting to us. One of the first scans that we did, which basically cut off 95% of the data, was whether they show up as significant in the lipid-related phenotype GWAS. We end up with something in the order of 30s for each of the cell types that we have. And of those, we're particularly interested in the ones that fall into the top orange circle, which represents HLCs. Remind you, we are interested in lipid-associated loci that has potential implications in cardiovascular disease effect. And so when we got to this level, and after finding a subset of those that also has statistical colocalization significance, my collaborators went on to do a lot of functional characterizations and validations, and I'll just tell you about some of the exciting ones that we found. If you want to know about the rest of them, there's a paper out there you can read. One of the things that have been an enigma for lipid-associated researchers was called CPNE1. To us, it was actually less of an interesting story because it not only showed up for HLCs, but it also showed up for iPSCs. And mind you, we are interested in the ones that we could prove HLC worthy. So we were looking for a story that would actually show up as HLC specific. But suffices to say that we have validated CPNE1 locus through both through statistical molecular in vitro, in vivo, and mouse model studies. So that was pretty cool. Another one that we were really excited about was called NGPTL3. Now I'm giving you a punchline here, but it was a liver-specific EQTL, and it showed up in HLCs in our cohort, but it did not show up in iPSCs as significant. As a matter of fact, I think the gene expression level was fairly low, so we couldn't get enough statistical significance out of them. So we looked a little closer to it. The particular what we call best SNP, or the minimal p-value SNP that we found in the region is long RS number, was an almost perfect LD with a GWAS locus that shows up for LDL cholesterol levels, triglyceride, and total cholesterol levels, which was very interesting. It has, relatively speaking, clinically significant change or predicted change in decrease of triglycerides, which again, it was very interesting to us. Now, if you actually look at the region, sorry for the small font, all the way to the right, we are indicating, I'm indicating with the orange arrow, the particular SNP that we found from our EQTL scan. There's a gene called DOX7 down the row. It's actually a fairly close LD with the particular SNP we found. But it happens to be an iPSC gene in our data set and does not show up in HLCs. And its role is not really liver relevant, to say the least. There's another gene in the region called NGPTL3. This was an HLC gene in our data set and does not show up for iPSCs, so that was interesting. And it has known regulatory role in blood lipids, which is, again, fairly exciting to us. So we looked a little closer. If you go to GTEx portal, you can plug in any SNP or any gene names, and then it will tell you whether that SNP or gene is included in the data set and whether that has any statistics they can share with us. This one in particular is showing bar plots of gene expression. On the x-axis, it's showing 44 different tissues that we represent in GTEx. On the y-axis, it's showing raw gene expression values that we quantified from whole RNA-seq data across 450 individuals in this phase of the data set. It suffices to say that you can't actually pinpoint liver because it seems to be expressed almost everywhere. If you look at NGPTL3, I don't even have to draw a box around it because it only shows up in liver. It doesn't show up anywhere else. So it's sort of like leaning towards that story that we are looking for. And it was not that hard to find, which was pretty amazing. And so I don't know how many of you would know much about CRISPR. I personally didn't know a whole lot right before I got involved in this study. So basically, my collaborators have done different levels of functional validations in order to prove whether the statistical significance exists and is detectable, molecularly speaking. One of which was knocking in a minor allele for the particular SNP of interest. Some biochemical assay, they call it piggyback. Anyway, they successfully knocked in a particular minor allele and created homozygous clone of the wild type, and have proven that GG being the wild type, AA being the homozygous minor alleles. DOC7 clone, excuse me. When you compare the wild types to the knock-in clones, DOC7 gene expression levels did indeed decrease. And on the contrary, NGPTL3 gene expression did increase. So that's just cellularly replicating our findings from the RNA-seq data. They also deleted the region using CRISPR-Cas9. It basically is, you can imagine like a molecular seizure that goes in, recognizes a site, cuts the part of the DNA off, and then it fuses them, pretends like nothing happens, and then you can run assays on them. For the knockouts, they used embryonic stem cells to be as close to stem cells as possible that we used to first study this. And it was fairly high efficiency, by which we mean that over 50% of the clones actually contained the deletion that we wanted to include. So that was pretty cool. And long story short, it basically has negative controls being negative controls, same between wild types and the knockouts. DOC7 again shows the decrease in gene expression, and NGPTL3 increase in gene expression. They also did alternate assay that basically showed the same thing. And I'm not showing this here in this talk, but they also have done mouse studies and basically validated the same things. And we were fairly excited about this. And we are carrying out some other functional assays that are related to lipid metabolism in particular, in addition to these functional assays that are proving the functional role of the particular SNP in the region for the genes that we are interested in. So in conclusion, we were fairly excited about the public databases such as GTEx that we are using as a crucial part of our study. So it's actually very difficult to get that size, that scale of data set by any means. That's why we require the consortium. But we do think that study-specific data such as ours did play a crucial role in discovery as a discovery cohort. And so we are working on actually building a computational pipeline that can be shared across many study sites that are involved in both of the consortia that will be able to facilitate this sort of computational pipelines that leads to candidate gene discovery using both public and private data. That's another story that includes Docker, et cetera, that I will not talk about today, but if you're interested, I'm happy to discuss it after the talk. And we do think that replenishable sources such as hiPSCs do provide a very, very valuable and convenient resource for a lot of the researches that we are working on. And it is certainly a lot easier to obtain than primary tissues. And so I would like to say that you're all welcome to go to dbGaP, download the data, and play with it a little bit, maybe compare it to your favorite other study example or model. So it was a work of a lot of people. I'm only listing a fraction of them for the space that is allotted on the slide. Primarily, a lot of the work has been done by Evanthia Pashus, who now works in Pfizer. She has done the iPSC cell generation and much of the molecular lab work. And once the data started coming off of the sequence machines was when I got involved. And you're seeing some of the results that I presented today. Thank you.
Let's now go straight to journal clubs because we don't have a lot of time, and we'll basically do that through as a journal club. Just